Hello students of class 10. Some of students you have doubt in dihybrid cross, but before starting dihybrid cross you must know about the monohybrid cross. So what is the definition of monohybrid cross? So it is a cross between two organisms of the same species which have single pairs of single character. That means you can see here it a monohybrid cross between a tall and dwarf plant. Mendel, who is the father of the genetics, that Mendel experimented uh, taking a single character of pea plant, tall plants and dwarf plant. So when and the alleles, what are the alleles of for the tall, that is capital T and capital T, and for the dwarf is small t and small t. Before this or in previous video I have explained this why TT is represent and small tt, that means always alleles in paired form, because it tall pure plant, that is TT, this gene is always represented in pair, one from female and one from male plant. So it is always represented in pairs. When cross between these two, so how the gamete is produced? You can see here the when gametes produce, these two pairs getting separated and single allele is present in a gamete, single gamete. So when cross between these two gametes takes place, that is called F1 generation. So what type of organism is produced? That is, all the plants are so tall, okay, and this is a hybrid, this is a pure form, this is also pure form, but in F1 generation what we get, that is all the plants are hybrid. And again Mendel taking these two hybrids once again, if we cross between these two, then the gametes produced in such case are capital T, small t, and this is called trait or character, and these two capital T, small t, these two are called alleles. Again gametes are produced capital T, small t, okay, these are the gametes. And when the similar type of gametes, if we cross them, then what would we get? So suppose TT, when these two gametes fertilize together to produce capital T, okay, then all plants are tall. Why? Because tallness is the dominant character. Again this T and this T, it is a hybrid. So capital T is present, then it is also a tall plant. Here small t and capital T. So it is also a tall plant because capital T is present here. And finally small t and small t. These two pairs makes a dwarf. So you can see here, and this is the F2 generation. That means this is first filial generation and this is second filial generation. So among all the plants, these three are tall and a single is dwarf. So you can see here the alleles are getting separated in this, and when these separated in for the second filial generation, this genetic recombination is capital T, small t, and finally the original trait, that is tallness and dwarfness, is again we can get here in the second filial generation. That is, this is a pure breed, first one, and this is again a pure breed. So we can say that the genes are not completely removed from the F1 generation, because this gene for the Daphnes is present inside it and getting segregated in here in the F2 generation. So this is called incomplete. So this is called laws of segregation. So what did I say? That as a result of gamete formation, or gamete contains only a single factor of a character, and it is therefore always pure for the characters. Because of it, the principle of segregation is also called principle of purity. That means when a cross between these two characters are taking place during the gamete formation, these two factors T and T randomly separated and at the gamete formation, and these two again combine together in the F2 generation, and it appeared that tall plant and dwarf plant, that is the formation of a pure homozygous tall plant and pure homozygous dwarf plant, clearly proves that it separates from the factor for tallness during the gamete formation. Coming together of two factors of dwarfness is possible if the gamete contains only one factor of character, that is, they are pure. So this is called laws of segregation. Okay, that means the during the first plant, that is this plant is again segregated in the F2 generation. So it is called laws of segregation, and this is the phenotypic ratio. That means the externally apparent plant are 3 tall and 1 dwarf, so it is phenotypic, and the genotypic ratio will be 1:2:1. That means 1 tall plant, and genetic in genotypic ratio we see the genetic combination, that is genetic combination, it is 1, this one is 2, and this one is 1. Okay. Now dihybrid cross. This is a cross, flow chart of cross between two characters, taking two characters of a single plant, that is tall rounded, one plant containing tall and rounded seed, and another was is dwarf and wrinkled seed. So the gametes formed in first filial generation, that means gamete produced by these plants are containing this TR, that is the genes will be capital T, capital R, that is for tall and round, and for dwarfness small t and small r, because this is the recessive character and this is containing all dominant character. And when these two gametes unite together, it produces the hybrid containing capital T, capital R, small t, small r. So all the plants will be tall, rounded. Why? Because tallness and the roundness both are the dominant character. And this is F1 generation. And when this F1 generation plants are self-pollinated, then the gametes produced by this hybrid is of four types, that is TR, TR, TR and TR. This is the male gametes, and you can see here the female gametes TR. And when the offspring, that is the child or the new organism is produced, it contains, when TR fertilizes TR, then the genetic combination will be this, and in this way this and this, in this way a flow chart is produced where phenotypic ratio will be 9:3:3:1, and the genotypic ratio will be much more. So we don't depict the genotypic ratio, only phenotypic ratio for the dihybrid cross. You must understand how these are appeared in F2 generation. These are the gametes containing TR as a genes. These TR are the genes for tallness and roundness, and these here tallness and wrinkled. This is the dwarfness and round, and this is dwarf and wrinkle. So these four types of gametes when unite together produces 9 tall rounded, 3 tall wrinkled, 3 dwarf rounded, and 1 dwarf wrinkled. So here the two factors of a character separated, that means here you can in the starting these are the two factors, tall round, okay. These TR are getting separated and paired independently. That's why the these tall wrinkled and dwarf rounded can be produced in the second filial generation. So this process of making an hybrid with new recombination, you can see here the tall wrinkled and tall rounded, and where is dwarf rounded. This is the hybrid containing new recombinations of genes, and this law is called independent law of independent assortment. So you can you have to read the definition of laws of independent assortment, and this flow chart you can take the screenshot of this. Take a screenshot for this flow chart. Okay, thank you.